Okay, friend, till now you are able to understand how these channels and how the channels work and how the membrane potential arise on a neuron membrane. Now the question arises how we measure that one. So how we measure that electric potential, electric current? For that purpose, you put a micro electrode, you will voltmeter. For this, there are two electrodes, anionic and ionic. So one electrode you put inside and another on the outside of the side, micro electrode, the micro electrode. So then the next we come to the graded potential. As you know, there is a small deviation from the resting potential which lead to the excitability of cells. So those cells, this excitability, their cells, there is a deviation, cells become polarized, means they are more negative inside than the in the resting potential. When the cells become membrane potential more polarized, that is negative, more negative inside, is termed as the hyperpolarization and the graded hyperpolarizing graded potential. And when the response that makes it less polarized, means less negative inside, is called as the depolarizing graded potential. A graded potential basically occurs when a mechanical and ligand gated channels in the dendritic cells or dendrite or cell bodies are open. Main cell bodies upper they form the presence of these cell body mechanical gated and this ligand gated channels are open, they lead to an certain disturbance in the they lead to the excitability of the neuron membrane. The amplitude of electric signal, a graded potential, is vary from the basically depend on the strength of stimulus. In the other word, you can say depend how many channels, how a stimulus, how many channels are open and closed in response to a particular stimulus. The opening and closing of these channels follow the specific ion across the membrane, which means this following there is a opening of flow of charge, flow of they form a current, localized current, which means they spread to the spread to the adjacent cells of plasma membrane in any direction from the stimulus. And the graded potential travels a short distance or die out from the or they die or lose through the membrane. When a membrane potential die out or from a throughout the both in a small distance of a membrane is known as the detrimental potential. A graded potential becomes stronger when it bind to the two same amplitude or different amplitude of graded potential. When they combine or it become the more stronger, and this is known as the summation, or this is known as the summation. Known as the summation. And when these there is a you see here, and when these two different potential with the summation, when two different potential but in opposite direction, they cancel each other. You see there's a gradient potential, opening and closing of channels are there. Okay, when the sum, the graded potential actually it's graded the amplitude or depend on the stimulus. As a high stimulus, you see there is a membrane difference is higher, and when Depending on the type of stimulus where they occur. For example, if the graded potential occur is neurons, in response a neurotransmitter is known as the postsynaptic potential. Postsynaptic potential, or if it occurs in a receptors, it's a sensory receptor known as the receptor potential. And you see in this process, the number of channels are open and closed, or in that particular response, they are the specialty of this graded potential, they are of short distance. But are those stimulus which are able to pass this information, they carry on, or they maintain a proper level of potential across the membrane, or they pass through them from one region to other region across the length of this axon. The events that occur during then the next phase of that potential, it's a accent potential. Second is the accent potential, or accent potential is a sequence of rapidly occurring events that decrease or reverse the membrane potential. As you know, there's a charges are changed across the membrane, or this actually accent potential try to restore the ionic concentration around that. During this event, it's a depolarization phase. Negative membrane potential become the less negative, reach to zero and become positive. During this repolarization, membrane potential is reached to a resting potential, that is minus 70 millivolt. The membrane potential of a may be an hyperpolarization state, in which the membrane is temporarily, inside membrane is temporarily become more negative. That's where the channels, during this action potential, the voltage gated sodium channels open fast, allow the influx of sodium ion and allow the influx of sodium ion into the cell, followed by the opening of potassium channels, which allow the outflux of potassium ion, which produce the repolarizing phase. For this depolarization, when it reaches to the minus 55 degree millivolt, it is known as the threshold. Those potential, those events, those action potential of neurons which reach to a certain 55 millivolt is known as the threshold. Means this is the threshold limit. Those action potential which depolarize the membrane, this up to 55 are passed on, otherwise reduce, or they are not get the action. The generation of action potential depend on whether a particular stimulus, अब एक stimulus कैसा है, it's able to bring the membrane potential to the threshold or not. If it is able to bring the threshold of 55 millivolt, the action potential occur, or if it did not occur, that is a sub-threshold stimulus. Several action potentials which in response to some stimulus is a suprathreshold. Those they form the throughout the depolarized membrane above the threshold. The characteristic is known as those response which form the suprathreshold, they are passed. Okay, suprathreshold, or which pass the threshold, they are passed. Or this phenomena in case of nerve is known as the none or all. Only those stimulus get passed. Yeah, they are produce action potential which have the minus 55 millivolt. During this, you will see the resting potential. These resting potential at resting this lead to the opening. There are events which occur during this uh, put, during this during the second phase of event of action potential is the in this it's a action potential, which means a rapidly occurring sequence of events that decrease and eventually reverse the membrane potential, depolarization, and eventually restore it to the resting state, that is repolarization. There is a list of events which occur during the propagation of a nerve impulse: the depolarization, repolarization, hyperpolarization, and opening and closing of these gated channels. In this action potential, the one is phase, the depolarization phase. In depolarization phase, the voltage gated channels, when a stimulus that cause the maximum to repolarize reach to the threshold, voltage gated channels, voltage gated sodium channels are open rapidly. Both electrical and chemical gradient favor the inward movement of sodium ion and outward movement of potassium ion, that is influx of sodium ion and outflux of the potassium ion, or the flow of these ions in actually the flow of sodium ions change the membrane potential from 55 millivolt to the 30 millivolt. At the peak, the action potential of this inside the membrane is 30 millivolt, that is more positive than outside. This membrane potential reach in actually with the, as you know, that during this depolarization, sodium channels are open. The sodium channel, voltage gated sodium channels are the three phases. One is resting, action, and again inactivation phase. As I discussed earlier, there's a opening of in a resting phase, there is a inactivation gate are open. In activation phase, both both the channels are open, activity channels are open, and in third phase, again the rest in inactive phase, both channels are closed. The threshold of these gated channels are activity stage, and the voltage gated channel, both activation and inactivation. As the more sodium channels are open, the flow increase, or this the more this flow of sodium ion increase, or in response to a opening of sodium channels, or this is known as the positive feedback mechanism for the membrane action potential. The second phase of this is the repolarizing phase. In this repolarizing phase, the sodium channels are inactivity, means they are closing, and the potassium channels are open, or this event occurs in a simultaneous way. The sodium channels are closing, and these lead to the again this more potassium channels are open, and this closing and opening of these channels, because of this there is a slight difference, lead to the repolarization phase of action potential.